My Grand Challenges Exploration project is about testing the idea that MTB is fundamentally different than other bacteria that we've known about. The most significant progress that we've made is, in fact, providing the first kind of biochemical descriptions of how TB metabolism really operates, and to discover that, in fact, it is fundamentally different. Based on these differences, we have reason to think that there are going to be specific targets that we can attack to kill non-replicating or latent TB.
The goal of the project is to make non-hormonal male contraceptive, but which can be practiced by women. And so we thought if you separated the sperm from the sugar, which it normally requires for energy, then you deny it the energy to reach the egg.
My exploration project involves actually the use of cell phones to do microscopy and related diagnostics in the field, such that you could do blood analysis, or you could look at malaria slides, for instance, in remote locations where there's nothing but just working cell phones and some gadgets that could be attached to the cell phones.
So my great idea for this project was to try and develop a protocol of vaccination by which I could instruct T cells to go to the organ that is most likely to be affected by the pathogen. So we are targeting our immunity there to make it 1,000,000 times more efficient.
The problem is that HIV infects people and it stays with them for their whole life. And the reason for that is that the virus actually goes into the person's genome, into their chromosomes, into their DNA. So what my project is to find a way where we can actually go into those chromosomes, inactivate that virus so that the cell's alive, the person's healthy, but the virus can't exist anymore. So the idea is now to take these proteins, they're called homing nanonucleases, and change what they do in nature. We're going to change them so that they find HIV sequences within the human genome.
So our great idea in mosquito defense and malaria mitigation is to stop mosquitoes from reaching human host through using optical barriers between the mosquito and the humans. The basic idea was that damaging or confusing the sensory system and the neural network behind would help us keeping mosquitoes away from humans. And these optical barriers work.
The goal of my Grand Challenges project is to try to develop drugs that are less susceptible to pathogen resistance. So we think about how to design nanometer scale drugs to bind more tightly to protein targets in a pathogen like tuberculosis, and how those binding interactions might be able to better withstand things like mutations. We were able to take resistance-compromised antibiotics and redesign them onto a nanoscale platform and show that they were highly active again. Grand Challenges Explorations mechanism, where you get $100,000 to test out some hypotheses, is really key because it really gets you going.
It's fairly difficult to do very high-risk projects. The exploration program is a great means to kind of try those difficult-to-test ideas and generate some initial results.
They did not want me to provide massive amount of data, but they were quite happy to fund the idea and let me provide proof of principle. I would strongly recommend to any scientist that is trying to do this kind of thing to go for this kind of funding.
I tell them to think of the best and craziest idea they have that they really believe in, that the idea that they know in their hearts could work even if everybody else tells them it can't. And that's the one they ought to be putting in. Because if there's any place where it's going to get a fair hearing and a chance to become reality, it's in the explorations program.